Attention to all participants. We will start our opening ceremony.
Good morning, Magandang Umaga from Indonesia, and best wishes to all of us. Summer course is an experience that will broaden our experience and our understanding and help us envision future career paths. We will embark on a series of visits to natural pharmaceutical companies during this program. The third ISCP serves as a dynamic platform to connect experts, researchers, and students from around the world. It aims to foster collaboration, exchange of knowledge, and promote sustainable practice of latest innovation in the use of natural products in pharmaceutical industry.
Greeting to audience, partner, and participants from Philippines and Indonesia. The Honorable Vice Rector of Universitas Pancasila, Muhammad Rizky Aldila, Bachelor of Law, Master of Notary. The Honorable Dean of the Faculty of Farmasi, Universitas Pancasila, Profesor Dr. Apoteker Dian Ratih Laksmitawati, Master of Biomedic. Vice Deans of the Faculty of Pharmacy, Dr. Apoteker Gresti Vinotori Swandini, Master of Pharmacy, and Dr. Apoteker Andri Prasetyo, Master of Pharmacy. The Honorable Chair Committee of 3rd ICP UP, Apoteker Muhammad Iqbal Julian, PhD. The Honorable Executive Director, DEXA Laboratories of Biomolecular, Professor Raymond Rubianto Chandrawinata, PhD.
The Honorable President Director of B Pharma Adiluhung, Dr. Sandi Klintang, M. Biomed.
The Honorable Lecturer from Kuliah of Pharmacy, International Islamic University, Malaysia, Professor Denny Susanti, and Professor Muhammad Taher Bahtiar.
And finally, all participating students of 3rd ISCP UP 2026. As the Master of Ceremony, my name is Azumi Sultanika and my partner,
Matthew Vincent.
Representing this committee of this event, we would like to say,
Welcome to the opening ceremony of this year's Summer Course 2026. Ladies and gentlemen, before we step up to the next agenda, let's sing together the national anthem of Indonesia, Indonesia Raya, and Hymne Universitas Pancasila. For all participants, please rise. For all the participant, please be seated. Thank you.
And now we would like to invite the chair of the committee, Apotheker Muhammad Iqbal Julian RPP PhD, to give us this opening remark to all summer course participants.
Okay, distinguished Rector of Universitas Pancasila, Professor Dr. Adnan Hamid, which at this moment is represented by the Vice Rector, Dr. Muhammad Rizky Aldila, Magister Konotariatan, respected Dean of the Faculty of Farmasi, Professor Dr. Apotheker Dian Meratih, along with the Vice Deans, Bu Kristi and Pak Andri, and Head of Study Program. Our esteemed keynote speakers, Professor Raymond Chandrawinata, Dr. Sandi Klintang MDOME, Associate Professor Dr. Denny Susanti, and Professor Dr. M. Taher bin Bahtiar. Honor Chaplain from Trinity University of Asia, Mr. Joe Tambis. Dear lecturers of the Faculty of Farmasi Universitas Pancasila, committee members, and all brilliant participants of this program. Good morning and a very warm welcome to the opening ceremony of the third International Summer Course in Farmasi, Universitas Pancasila. It is an absolute honor and privilege to stand before you today as the chairperson of this event. We are deeply grateful for the presence of our university and faculty leaders, our distinguished global expert and participants who have gathered here today. This year, our summer course carries a forward-looking theme, Bridging Traditional Wisdom and Advanced Biotechnology, the Future of Global Pharmacy. As the landscape of global healthcare rapidly evolves, we are challenged to look both backward and forward. Traditional medicine holds centuries of proven wisdom, while advanced biotechnology offers the precision and tools to unlock its full potential. Over the next few days, this program is designed to be a collaborative melting pot where these two worlds meet. Through insightful lectures, discussion, and cultural exchanges, we aim to inspire the next generation of pharmacists to innovate for a healthier world. I would like to extend my heartfelt gratitude to the management of Universitas Pancasila and the Faculty of Pharmacy for their endless support. To our esteemed speakers, thank you for sharing your invaluable expertise. A special welcome goes to Mr. Joe Tambis and student delegation from Trinity University of Asia. Your participation beautifully embodies the international spirit of this event. Lastly, my deepest appreciation goes to the organizing committee, whose hard work and dedication have made this day possible. To all participants, I wish you an enriching, inspiring, and memorable experience throughout this summer course. May this event foster new friendships, spark brilliant ideas, and broaden your academic horizons. To officially close this opening remark, allow me to deliver a traditional Indonesian poetic gesture, pantun, in English. Okay. Green leaves whisper a secret of the past, while modern science lights the way ahead. Welcome to this course, May Friendships Last, as new path in global pharmacy we tread. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you so much, Mr. Iqbal, for your wonderful speech.
Next, we would like to invite the Dean of Faculty of Pharmacy, Universitas Pancasila, Professor Dr. Apoteker Dian Ratih Laksmitawati, Master of Biomed, to give us the opening remarks to all summer course participants.
Tes. Assalamualaikum warahmatullahi wabarakatuh. Good morning, distinguished guests, ladies and gentlemen. The Honorable Dr. Muhammad Rizky Araldila, Vice Rector III for Student Affairs and Alumni of Universitas Pancasila, the Vice Deans of the Faculty of Farmasi, Universitas Pancasila, our distinguished representative from Trinity University of Asia, Philippines, our distinguished speaker and invited guests, respected lecturer, dear participant from Indonesia and the Philippines. It is my great pleasure to welcome all of you to the third International Summer Course in Pharmacy ISCP 2026, hosted by the Faculty of Pharmacy, Universitas Pancasila. First of all, I would like to express my sincere appreciation to Dr. Muhammad Rizky Aldila, Vice Rector III for Student Affairs and Alumni of Universitas Pancasila for joining us today. I also extend my heartfelt gratitude to our colleagues from Trinity Universitas of Asia, Philippines, for their continued partnership and commitment to strengthening international collaboration in pharmacy education and research. This year theme, Bridging Traditional Wisdom and Advanced Biotechnology, the Future of Global Pharmacy, reflects our shared vision of integrating traditional medicinal knowledge with modern scientific innovation through cross-country and cross-disciplinary collaboration. we can contribute to developing innovative sustainability and accessible healthcare solutions for the global community. During this program, participants will engage in lecture, laboratory, and fieldwork, interactive discussion, and cultural activities. These experiences are designed not only to enhance academic knowledge and professional skills, but also to foster international friendship, mutual understanding, and future collaboration. To our participants, I encourage you to make the most every opportunity to learn, exchange ideas, and build meaningful connection with fellow students, academics, and professionals from diverse backgrounds. I also hope you will enjoy the warmth of Indonesian hospitality and experience the richness of our culture during your stay. Finally, I would like to express my sincere gratitude to our international partners, distinguished speakers, the organizing committee, and everyone who has contributed making this program possible. I hope this summer course will inspire new ideas, strengthen international partnership, and foster lasting collaboration that will advance pharmaceutical sciences and global healthcare. Thank you for your participation, and I wish you all a productive, enjoyable, and memorable third International Summer Course in Farmasi 2026. Wassalamualaikum warahmatullahi wabarakatuh. Salam Pancasila.
Thank you, Prof. Dian, for your wonderful speech.
Our next agenda, we would like to invite Vice Rector of Student Affairs and Alumni of Universitas Pancasila, Dr. Muhammad Rizky Aldila, Bachelor of Law and Master of KN, to give us the opening remarks to all summer course participants.
Tes. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi semua. Salam sehat dan salam Pancasila. Terima kasih telah hadir semua dan izin membacakan sambutan ya yang diamanahkan kepada kami, dan salam hormat dari Pak Rektor Profesor Tan Hamid menyampaikan pidato dan sambutannya. Okay, thank you for all respected Dean of Faculty of Pharmacy, Professor Dr. Apoteker Dian Ratih, LM Biomed, along with the Vice Deans and Heads of Study Programs. Our distinguished keynote speaker from Raymond Chandawinata, thanks Prof. Dr. Sandy Kelintang, M Biomed, Asok Prof. Dr. Denny Susanti, and Professor Dr. M. Tahir bin Bakhtiar. Another lecturer from Trinity University of Asia, Mr. Joe Tambis, thank you for coming. Extended lecture committee members and all brilliant participants from around the globe. Good morning, peace and prosperity be upon us all. It's a proud and honor for me, Vice Rector of University of Pancasila, to stand before you today representing our lecturer, Professor Dr. Tan Hamid, on his behalf and on behalf of the entire university leadership. I extend our warmest welcome to all of you to the third International Summer Course in Farmasi, University of Pancasila. Universitas Pancasila takes a great pride in the Faculty of Pharmacy for consistently fostering global academic synergy. Events like this summer course are vital milestone in our journey toward internationalization, cross-border collaboration, and research excellence. We are especially delighted to welcome our delegates from Trinity University of Asia, led by Mr. Joe Tambis, Thank you. Your presence highlight the true spirit of global partnership that this university highly values. The theme chosen for this year's program, bridging traditional wisdom and advanced biotechnology, the future of global pharmacy, is exceptionally relevant. We live in an era where modern healthcare must look forward without forgetting to the foundation of the past. Nature and traditional knowledge hold fast, update secrets for healing, but it is through the lens of advanced biotechnology that we can validate, standardize, and scale this discovery safely for the global population. Through the expertise of our outstanding speakers, Prof. Raymond, Dr. Sandy, Prof. Doni, and Prof. Taher, I am certain that the next few days will provide you all with groundbreaking insight. I encourage all participants to actively engage, challenge ideas, and build lasting international networks. You are the future innovators who will shape global pharmacy. I would like to express my deepest appreciation to the Dean of Faculty, Prof. Diarati, and the entire organizing committee for their meticulous planning and dedication to making this event reality. By pronouncing Bismillahirrahmanirrahim, and on behalf of the Rector of University of Pancasila, I officially declare the third International Summer Course in Pharmacy open. Thank you. So, thank you very much. Have a productive and inspiring summer course and enjoy your time on University of Pancasila. Wassalamualaikum warahmatullahi wabarakatuh. Salam sehat and salam Pancasila. Yeah, thank you.
Thank you so much, Mr. Rizky, for your wonderful speech.
And for our next agenda, we would like to present an amazing traditional dance performance by UKM Besi FFUP. Renggong Manis is a traditional dance inspired by the rich cultural heritage of West Java, with graceful movement and lively rhythms. This dance symbolizes harmony, hospitality, and the joyful spirit of Indonesian tradition, reminding us of the importance of preserving our cultural identity. So please welcome UKM Besi FFUP. What a great performance from the CFFUP. Now, let's move on to the next agenda.
Before we move on to our first lecture, we would like to invite the Vice Rector, Mr. Muhammad Rizky Aldila, the Dean Faculty of Pharmacy, Professor Dian Laksmitawati, Vice Dean of Faculty of Pharmacy, Dr. Apotheker Gersti Pinotori Swandini, and Dr. Apotheker Andri Prastio. And our today's speaker, Professor Raymond Chandrawinata and Dr. Sandi Klintang, and all the lecturers that are present in this event to come up to the stage to take a picture together.
For the documentation, please. So for the picture, it's formal pose. Take picture in 3, 2, 1. Okay, another pose, Pancasila greetings. 3, 2, 1, and the last pose, freestyle. 3, 2, 1. Okay, thank you, and please be seated. Okay, I have some information for participants. So we will have prize for those who ask question during each session. So make sure to pay close attention.
Okay, for our first agenda, we'll be sharing materials which will be given by Professor Raymond Chandrawinata, Executive Director of DEXA Laboratories of Biomolecular Science. To ensure the session proceeds smoothly, we're honored to have Mr. Zainur as our moderator. Before inviting Mr. Zainur, please come up to the stage.
Okay, everyone. Good morning. Assalamualaikum warahmatullahi wabarakatuh. Welcome to the third International Summer Course Farmasi, Universitas Pancasila 2026, under Grand Theme, Bridging Traditional Wisdom and Advanced Biotechnology, the Future of Global Pharmacy. Honorable Rector, Universitas Pancasila, represented by Mr. Rizky Adila. Respected Dean of the Faculty of Pharmacy, Professor Dr. Dian Ratih, Dr. Gresti, Dr. Andri, and respected lecturer, faculty member, and all the academic community of Universitas Pancasila. A warmest welcome to our distinguished guests, the delegation from the Philippines, Trinity University of Asia, and my friend, Mr. Joe Tambis. Welcome to Pancasila. And all the students and participants for this summer course. Hello, students. Good morning to you. Okay. For this session, on this Monday, we gather discuss a highly critical topic for this session about National Pharmaceutical Independence Blueprint, integrating biodiversity with modern clinical science. Today, we recognize that the core issue in the global pharmaceutical system is not about technology. It is extreme inefficiency in R&D and unmanaged biological complexity. We will explore how AI, or artificial intelligence, is shifting pharmaceutical R&D from costly trial and error to data-driven predictive system. To break down this blueprint, we are deeply honored to have the global expert, Mr. Professor Dr. Raymond R. Chandrawinata, PhD. Good morning, Prof. Okay, Professor Raymond is truly unique and visionary leader, a biomedical scientist, with the CV, a legal scholar, and theologian who masterfully integrates science, law, and ethics to design the future healthcare system. His philosophy reminds us that science provides knowledge, and law provides justice, and ethics provide direction. Okay. He earned his master and PhD in biochemistry from University of California, Riverside, followed by postdoctoral fellowship in molecular pharmacology at the University of California, San Francisco. He also advanced research and He has published over 1,350 Scopus Index scientific papers and hold more than 30 international patents, mashallah, in US, Europe, and Asia. And he was ranked in first top scientist in Indonesia by AD Scientific and top 10 influential leader in healthcare. And so the prestigious OBB Award in Medicine and Biotechnology. Okay, currently, Professor Raymond served as Executive Director of DLBS, right? DLBS or DEXA, Laboratories of Biomolecular Scientists. Today, he will guide us through the essential blueprint of integrating biodiversity. Please join me. One plus for Mr. Professor Raymond Chapta. The floor is yours. Siapa bang? Oh, boleh.
Thank you, Pak. Sorry.
Zainur. Zainur. Yes.
Thank you, Dr. Zainur, for your kind introduction. I would like to acknowledge the Vice Rector, Dr. Rizky. Yeah, thank you. Very nice gentleman I just met this morning, but we share the same vision about law. So sorry. But the newest dean, faculty of Farmasi, Dr. Dian Ratih, Professor Dian Ratih, thank you so much. All the colleagues that I've seen and I met a long time ago, so we meet again today. Yeah, Dr. Iqbal, yeah, and also my colleague here. I've known him for the last 26 years, Dr. Sandy Kilin. If you want to learn about stem cell and all the derivatives, he is the guru. So you're very lucky to have him today because Indonesia does not have too many stem cell guru, but he's one of them and he's the leading stem cell researcher. And his business acumen also. Thank you so much. Yeah. Thank you, Prof. Dian, for inviting me. Slide, can I have? Slide? Okay, so again to students from Philippines, I'm so sorry. I just learned that I have to speak English and I gave my slides in Indonesian. So hopefully, if you follow me, then you will know what I'm talking about, okay? Thank you. Good, cool? Cool, okay, good. So today, I'm not going to be talking about his expertise stem cell. I'll be talking about natural products. Yeah.
Iya.
Apa di sini?
Boleh.
Penggema soalnya.
Gemma ya. Mas, dikurangin gemmanya.
Okay, thank you. Much better. Yeah. Okay, so I'll be talking about natural products and the reason why natural products has been neglected, especially in Indonesia. Oh, why do you say so? We are the number 2 and number 3 biodiversity in the whole world, even after Brazil. Apparently, as you will see, yes, we have 3 kinds of natural products. One is actually jamu, secondly is standardized herbal, and number 3 is actually in phytopharmaca. So in the Philippines, so my friends in the Philippines, there's only one registration, which is natural traditional products, natural traditional drugs. There's only one registration, which is considered to be drugs, not considered to be supplement, not considered to be jamu. Yeah, so in the Philippines, because I've shown you later that we have some trials also done in the Philippines. Yeah. So slide. So today we will be talking about natural products. Oh, lewat sini bisa. Okay, so you will see here that here in Indonesia, the market is still underdeveloped. Soon it will be surpassed by my friend here, cell therapy. Because why? Because we have all the jamu and everything, but yet you can see here, TCM, which is traditional Chinese medicine, now has $300 billion. India 50 to 100 billion US dollar. Indonesia is 3 to 5 billion, which is only like a fraction. How do we do? Why are we doing that? Yes, Indonesia, we're number 2 and number 3 biodiversity in Indonesia, but yet the value of our traditional medicine is still much lower. Then now becomes global medicine, which is TCM and also Ayurveda. In the Philippines, the value is much higher than this, I tell you the truth. And they don't have the biodiversity, but the value is much higher than this. I tell you, it's about 10 billion US dollar. Yeah. So we have a lot of things to catch up. This is what I'm going to talk about. Yeah. Now, if you're talking about phytopharmaca, everyone so glorifies phytopharmaca. That's the highest, hard to make. But don't worry, the market is still small. 12.1 million. Yeah. This is billion rupiah. 2024. The government acknowledges it goes down 4 billion rupiah to 8 only September last year, 2012. Means the market of biodiversity in Indonesia is not being recognized by the government. Only in Jammu, which people take it. But if I ask in this room, who is doing research on traditional medicine, Jammu or phytopharmaca, maybe everyone will go up in hands. But yet you can see here, the government still does not appreciate what you have done. What you have done, even if you have a good, wonderful products. Even phytopharmaca is going down, and you can see here, this is only a fraction, 100, much less than TCM. So we have a lot of works. We have a lot of homework, not only in the lab. My colleagues here, I tell you, we're not only in the labs, but at the policy level. Pariski, that's why I'm learning law. That's why I study law. I have to make sure that all my colleagues who have done a lot of research on this area will be awarded by the government, which have not now. The only thing that we have to do is only through law, through legislation. Okay, next. But do not worry, we're going into there, yeah? Because now we have a lot of potentials, we have a lot of ways to do that. This is actually therapeutic, kedaulatan therapeutic. What is it in English? Kedaulatan. No, yeah, not only independence, but sovereignty. Sovereign, yeah? Sovereignty of therapeutic. Using what? Kekayaan ekologis. Ecologic richness. Meaning using herbs, meaning you can do as an extract, as you can do as a supplement, whatever you want to do. But we have to do that. We have to make sure that now, if you ask, if we have that war just now, Indonesia import a lot of API, which is bahan baku, which is the raw material for drugs. How can we not use them coming from biodiversity? Sometimes we have to question the motivation of the Ministry of Health because they don't want to switch from chemical into herbal. So let me ask you now another one. In the Philippines, this is a different story. But here in Indonesia, we are all the member of BPJS. Yeah, we have the card. If we get sick, we go to doctor, which is covered by BPJS. Now, if I ask a drug coming from biodiversity in our formularium national formulary, can we get herbal drugs? No. Indonesia is one of the few Asian countries, I've done studies, which prescribe herbal drugs into the citizenship. In the Philippines, they do. They do. Now, Philippines is actually following us at BPJS, but they're allowing the citizens to prescribe via the herbal drugs. We cannot. So that is how much our government appreciates us as investigator, as a researcher for herbal drugs. Yeah. But again, we will talk about the different technology we use. But at the end, we want to have an integration of system kesehatan, integration of health system. Not only somebody prescribed using the chemical product, but also they have to be able to get the rights. As citizens, they have the rights to get prescription based on herbal drugs, which is now the rights is deprived in this country. Another one is deprived. Narcotics. Dr. Sandy, you know, yeah? If somebody has a lot of pain from cancer, they cannot get narcotics because the law does not allow us to get prescription medical narcotics to many patients. They are so worried that this will be abused. So there's only 1 or 2 abusive people, But the whole thing, patient who have rights did not get the medication, the correct, which is narcotics, oxycodone, anything. Yeah, it's not. So they're depriving again. So not only depriving the rights from the herbal product, but also narcotics. Okay. Now we use empirical evidence, but we don't have to do that. In order to increase the knowledge and also the amount of science, we use therapeutic with evidence. So we have to use evidence-based medicine, but on the herbal products. How do we do that? Now, you know, here in University of Pancasila, Faculty Farmasi, of course, we have to do animal studies. We have to do toxicology studies. We have to do pathophysiological studies. We have to do cell studies. Using what? Cell. What is a cell being used for? Only to find mechanism of action. So I propose anyone who wants to develop a phytopharmaca, the first thing that they have to do is actually hewan study, animal study. So we have to get the good material animal studies, and then from there we take it forward into the cell studies. The cell is only used for the mechanism of action of the herbal drug that we're using, and then we can go a bit further into in silico. So don't start with in silico. That was my past mistake. Oh, in silico, yeah, it's a brand new and everything, but it does not show pathophysiology. It's only mimicking what's inside the cell. Do we need to do that? Yes, we need to do that, but only do after the animal study, and then you do the cell study, and then you do silico study. So I give you the clue. So essentially from extraction, when you have the extract material, I'd like to use not the whole extract, but I'd like to use the fraction of bioactive fraction. What is bioactive fraction? So if you have this plant, you extract it 2 or 3 times. The first one using polar, second one using non-polar. This is an example. Third one, extracting it with polar. Then you have a bioactive fraction. What is a bioactive fraction? It's a fraction, meaning, for example, like me, you take only the arm of all the component in my arm. This component of my arm will have a different pharmacological action than if you take the leg, basically. So you take the different several times of the fraction, then you get a good pharmaceutically distinct products, pharmaceutically distinct. So from there, then you do chromatography profiling. Now we use LCMS and GCMS. And then you do chemometric analysis just to make sure that what you have the fraction, it has the different chemical synthesis. So chemometric is a way, yes, yeah, thank you. Chemometric is a way how to segregate the different types of components into one area. So we need to do that. Otherwise, you do not know what is inside your extract or bioactive fraction. And then from there, you find one of them becomes chemical markers. This is very important because standardized herbal and phytopharmaca need some kind of identification, need some kind of standardization. So where do we get it? From one of the chemometric analysis, and we get the chemical markers, which become our standard. This is very it's needed for the production of the finished product because you have to standardize. You have to standardize from the raw material into the finished product. If you don't have good standard, then you don't have something that you can quantify as marker. Yeah. So this is how we do it in the industry. So this is the extraction, and then using the bioactive fraction, and then you do chromatography using the advanced analytical techniques, and then you do chemometric, and then standardize using one of the chemical marker. Yeah. So after that, then you do chemical. This is the network pharmacology. This is in silico. Yeah. So this is you do at the end, like I said. The reason why we do at the end, because if you do it in the beginning, this does not apply, does not show what good it is in the pathophysiology. That's why I always urge you to find an experimentation in laboratory animals. Starting from there, then you go to the cell, and then you go to in silico. Now in the silico, now we do network pharmacology. Essentially, we want to find the protein-protein interaction. We want the extract interaction. We want to have the bioactive fraction interaction with what target which being used as the alleviation of the pathophysiological problem. So that's why this is good, but do it at the end. Yeah? Don't do it in the beginning, because otherwise you do not know whether this, if you do it in the beginning, you do not know whether, number one, the compounds found in the network pharmacology is actually something that we have in vivo, may not be the same. Secondly, the target in which we find in the network pharmacology may not be exactly the same as what we found in vivo. So I always like to do it backward, from animal to cell to in silico system. Yeah. So essentially, now what we're doing is a discovery. This is about the biotic fraction, and then the biology. The biology could be coming from the animal, and then the cell, and also in silico, this is the biology. And then development, because your industry, we have to do formulation development, standardization for the manufacturing. Yeah. So our experience, you have the pharmacy students, formulation is very important. Formulation in the herbal, it's easy as you think. The reason being is this: if you formulate one compound, for example, paracetamol, everyone can do that, and everyone can, or ibuprofen. That's easy to formulate. It's only one compound. Remember, in a fraction, it contains at least 50 to 100 compounds. And there are a lot of interactions within that particular extract or bioactive fraction. So to make them, we make sure that we have standardized every single extract, every single bioactive fraction. And number two, that's a standard. Number two, we have a good formulation that is stable. Yeah, that will not be easy. In our experience, once you have a bioactive fraction, sometimes it has a lot of water. So how are we going to formulate it if it's got a lot of water? Hygroscopies. And then on the other hand, it's got a lot of fat, because we bioactive fraction, only fraction of certain compounds. So that's why it's not, if you take the whole fraction, the whole extract, maybe it's easy to formulate. But with the whole extract, you don't have a good pharmacological distinctiveness. But with bioactive fraction, for sure, you will have good distinctiveness, because we take only the compounds that has some pharmacological properties. But again, the caveat of having the bioactive fraction is sometimes it's not easy to formulate because of the technical challenges, because of the physical-chemical properties of the bioactive fraction, which doesn't allow us to formulate. So in the industry, if you find a good bioactive fraction or a good extract, always we want to have a highest yield with a lower COGS, lower cost of goods sold, lower cost of development. Now, that's another challenge. Finding good bioactive fraction that has good property in the pharmacological property, it has good safety, it must have high yield and low COGS, low cost of development. That's why making herbal drug is much harder than if you make a generic product. Because generic product, you can see USP, you can see Pharmacopoeia 6 from Indonesia, you can see European Pharmacopoeia, anywhere. But in herbal products, especially if you isolate from your own plant, yourself, we don't have Pharmacopoeia, we don't have the standard to develop those products. So that's why the competency of the pharmacist will have to be very knowledgeable and high-notch. Secondly, nowadays, we have a lot of analytical activities in which allow us. So some of you will become analysts if you don't work in the clinical setting, work in the industry, you work in QC or you work in R&D. Some of you will become the analytical technician, analytical pharmacist. Nowadays, analysis is higher. So in our company, we have 3-to-1 ratio. 3 analyticals to 1 formulator. We need more and more analyticals. Soon, maybe, probably. The global standard is 3-to-1. In the future, maybe 5-to-1. Why? Because we have a lot of impurities. The impurities sometimes something that we don't know, especially from plants. What kind of impurities do we have? We're not only talking about the metal, No, we're not talking about biobacteria, bioburden, but we're talking about impurities that comes from our extraction, coming from fraction. This is something that only can be done if you have a knowledgeable analytical. And then from that, we use a different LCMS, whatever GCMS, to make sure that our extract and our fraction will be quantitated. Yeah. So again, we use this different development system, and the newer generation of pharmacy student in the analytical world, they have to know not only it's already old. LCMS, even LCMS sometimes. Because some of us who work as a pharmacist in the bioanalytical field, you're doing bioequivalence study, you're doing pharmacokinetic study, you will need to know bioequivalence using the LCMS, for example. Yeah. So that's the so what's needed here. So about artificial intelligent pharmacy, as
Senior. Yeah.
I wrote a book on this. How do we discover new drugs using artificial intelligence? So, Prof. Dian, I will be glad to supply to Fakultas Farmasi. Yeah? But I wrote everything here about this is the decision architecture and transformation of system. Why is AI not only a tool? AI is a tool only for the technician, but for somebody who actually discovered the managerial discovery, as a discover, AI works differently. We have to make sure that AI is tools as a tool, use it very, very properly. Number one, why are we using AI? Number one is not because of technology, but because of the structural system. Right now, in the pharmaceutical field, even the Pfizer, the Sanofi, I've done the study, global multinational companies, their R&D system is very inefficient. This is why, very inefficient. This is why they have only 1 or 2 products per year. But now, the powerhouse for R&D is not in UK, not in Europe, not in US, but in China. So in your generation, we will have a lot of new chemical entities from China. And the reason being, they use the AI system very consistently. So AI as a way of life, not as a tool. So when you talk about in the R&D, AI is a way of life, meaning the whole system will have to be revamped. Working, the process system has to be AI based. That's what they do in China. So that's why a typical company in China will have about 3 or 4 NCE every year. Just remember, they have thousands of startup AI companies in China nowadays. If they come into the studies and become the product, we will have about at least probably 300, 400 new chemical entity per year from China. Yeah, maybe the regulation it's only for Chinese Chinese dossier, but still, the world needs a lot of new products. The world needs a lot of new chemical. For example, semaglutide. Semaglutide. Ozempic. Ozempic, yeah? Yes. This is for diabetes, for cardiovascular. It also for the obesity. But in China, people making semaglutide, that's easy for them. Very easy. They probably have about 300, 400 brands for semaglutide in China. Even Monjaro, which has just been Monjaro just launched here a month ago. It's been launched in China probably about 2 or 3 years ago. In the US also, 2 or 3 years. But they have a lot of Monjaro-like product already. But now, they're not satisfied by having semaglutide or tirpacetide. Yeah, tirpacetide is the brand, the generic name for Monjaro. They have GLP-1 agonists. Every single company that I visited have their own GLP-1 agonists. So, if you ask me, how many GLP-1 agonists in the market, maybe within 1 year or 2, I would say probably about 50. Not only coming from Novo, which is the semaglutide, or MEP coming from Wegovy, Monjaro, but the Chinese company are already having like 40 to 50 of this NCE of GLP-1 agonists. So peptide is another frontier. So in Fakultas Farmasi Pancasila, if you know how to formulate peptide, very good. Into injectable, especially we have the long sustained release peptide. That's very, very good. You will have a good formulation because everyone is moving from small molecule to peptide. But today we're talking about natural product.
That's natural.
So essentially, everyone, the AI is not only being used as tools, but revampment. The whole thing, we have the refreshment of system. Yeah. So which one are we doing? You have to know these three: generative AI, large language model. This one is there's a few technologies for this. Is for drug discovery. For machine and deep learning, we have to use also prediction precision for the property of molecules. Yeah, we use this not in the discovery. We use this in the industry as formulation. So start formulation, we have to start using the AI already. Especially we want to predict whether this will become PE or not. Let's start in the AI, machine and deep learning. And then agentic AI. This is agentic AI will help the brain how to create a good mentoring and for the automation of the R&D for autonomics. So this one also we use for simulation of clinical studies. Because clinical study can cost a lot. So that's why we use the AI, agentic AI, to at least prescribe and predict whether this will be meaningful in the clinical studies, in the clinical trial. Otherwise, it's a waste of time and it's a waste of money. Yeah. Okay. Okay, I'm going to skip this. Yeah. So this is an example. We have a product called Fiprolin, which is coming from Valeria macrocarpa, trans god, or otherwise known as Mahkota Dewa. Now, in the Philippines, we don't have the Mahkota Dewa, but we have in Papua region, which is this is to the east of Indonesia, in Papua. They have a lot of exotic fruits, and one of them is Valeria macrocarpa. So I'm just going to give you an example how we started with the mice study. This is hepatoprotector and lowering on the MAFL, which is metabolic associated, heart, liver disease. And then we went to the cell from the which is animal to cell, and then going to in vitro. So this is the result of the in vitro. So we already published this. Okay. So here, MAFL burden. I'm not going to talk about this. This is actually hepatic cirrhosis and fibrosis. Because of the inflammation, the liver cell will become cirrhosis. So we use this Valeria macrocarpa, but we use not the whole fruit, but we make biostandardized fraction. Then we call proliferanol. And then we develop into drugs with the clinical trial and everything. It's called Fiprolin. So these are the component that we know that coming from chemometry and also coming from the in silico system. These are the one that actually binds the receptor in the liver, and we know the receptor in the liver already. So next, now here's the in silico validation, and here's the in vitro, within the cell, in vivo, PTGS2, SERT1, GSK3B, RELA. These are the actual receptor within the cell itself. How do we know? We use the in silico study using network pharmacology. And it turned out that many of this is actually belonging to this anti-inflammatory, anti-apoptotic, antioxidant, and also metabolic regulation. And also, this is what we want, is the insulin sensitivity, glucose homeostasis, and lowering the hepatic steatosis and lipid accumulation makes it a very good MAFL product. And if you ask, what is the chemical drug that can be used for MAFL? None. There's no product coming from chemical world. So that's what we have to find in the natural product. So this is an example how, if you want to find a new drug product, do not find and mimic whatever it's in the chemical field. You have to find a new niche. Otherwise, your invention cannot be sold. Number two, all the products now, when they enter the e-formularium, the cost is very low because the government put a lot of cost. Metformin is only $50, 50 rupiah. For example, sulfonylurea, 50 rupiah. A new drug like SGLT2 agonist just now, empagliflozin, is only 300 rupiah. But if you find a new extract mimicking metformin, mimicking sulfonylurea, government will not purchase because it guarantees it will be much higher than metformin typical generic products. So that's why if you work in this area, you have to find a niche in which there's no chemical drugs. Then the product sells because there's no substitution. And then you can sell it with the higher amount of money that you can sell. Because nobody can substitute it with any product. So now this one, I have to confess, we have done this in the Philippines. My friends, Filipino friends. This is done in the Philippines by clinical doctor in the Philippines, Dr. Yalung. You find it in Metro Manila, Dr. Yalung, my friend, has done this. What he did is he is actually somewhat like a surgeon, but take care only of diabetes foot infection. That's what the clinic works with. Yeah. So this one is you see ulceration here, and what he did is actually they gave dissolve, which is phytopharmaca. This is coming from the earthworm, bioactive fraction in the earthworm, and bioactive fraction from the Chana striata, which is fish. You have also in the Philippines, Chana striata. And then lo and behold, the patient, look at this. They are consult, you got a lot of nephrotic lesion here. And starting, it becomes drier. And finally, within day 60, 2 months only, the foot becomes normal. The foot becomes like yours. So this is an example. I want to give this to show you that people claim, oh, natural product, it doesn't produce a good clinical effect. That's no chance. If you have a good clinically designed from the beginning chemical product, it's as good as chemical product. What would you have done this? Probably people will use antibiotics, for example. Yeah, we use growth factor, for example. But we don't have to use that because we use only dissolve, which is coming from the earthworm, and then also the inbumin, which is coming from the fish. Then the fish, the food can heal itself. You cannot dispute the result of the chemical, the result of natural product if designed properly, using good pharmacology, using good animal way of doing things, and clinical trial like this. Yeah. So this is an example. If you work in chemical in natural field, do not get despair because your product is not as good, not as efficacious as chemical product. No. Natural product, again, I will have to repeat, if you design it properly from the beginning, you have the right bioactive fraction, you have the right mechanism of action, you show pathophysiologically it works in animals, you show in silico, and then when you take it to human being, this is the result. So nobody argues with me anymore. A lot of doctors, oh, that's natural products. It used to be, they always disputed me. Oh, what's your product? Phytopharmaca. Ah, I have chemical product. Your natural product will be loosened. Your chemical product will not work in my patient. No. I have shown over and over. We as the investigator, we have the researcher of natural product. If we work properly and design properly, it's as good as chemical product. And sometimes it gets even better. Yeah? Okay. So here's what we need to do. Yeah. So here's biodiversity. You have a lot of things. Yeah. And then research molecular. And then transition means from lab to bedside. Sorry, from bedside to lab, and then going to bedside again. Yeah. So we always have to embrace the clinical pharmacists. We have to embrace with doctors who's done the studies, super specialty of the doctors who have done the study, like Dr. Yalung from Manila, who have done dissolved in albumin patients clinical trial. And then from that, you will have commercialization. Yeah. So Here's again, my complaint is Indonesia does not get respect. Phytopharmaca Indonesia does not get enough respect from the government because it's not into the formularium. Once it gets into formularium, it takes only one. We have to change kementerian. We have to change keputusan menteri, yeah, Capman, yeah. If that, then we will have a good system using the natural product, not only in the everyday use like jamu, but also in the fast-case, so health facility, wherever the doctors practice, yeah. Yeah, exactly. Yeah, so this ketiadaan, it's not in the BPJS. Yeah. So in order to do that, again, we need regulation for phytopharmaca, which is not happening now because the Menteri, the current minister, does not want to change that. Capman Case number 8, 2024, that has to be changed in such a way that allow the natural product incorporated into Formularium National. And that way, our research, whatever we're doing, will be valued by the government. Our research will be respected by the government. It doesn't end up in thesis. It doesn't end up in patent. It doesn't end up. But we need more products coming from the natural product in which you have done a lot of works. You get grants, you do research, you get a good material, and then you formulate into good product, and yet it's not being used by our own government. Very sad. Yeah? Hopefully in the future, the government realizes rather than importing so much chemical APIs into the country, let's use whatever we have in our backyard. Yeah, and if that's the case, then we as a researcher will be much valued and will be well regarded. Yeah, research doesn't go away. Research will still be there. Okay, thank you so much. Hopefully it will make sense, and welcome for the questions.
Okay, thank you very much for Mr. Raymond. Thank you. Absolutely, about our topic today is very improvement, yeah. About we know about how the natural product as good as chemical product, and how we can improve our extract or biofraction for this ecosystem. Yeah. Maybe our government can appreciate us as pharmacists as and respect for our work, yeah, and maybe allow to Formularium Nasional, yeah, Formularium Nasional. Go. Okay, ladies and gentlemen, yeah, okay, maybe we will now open the Q&A session. Yeah, if you have a question, maybe please raise your hand and state your name and institution and deliver your question directly to the point. Okay, maybe one or two participants can ask and raise your hand. Raise your hand. Sena? Maybe from Philippines, any question? About this topic is very interesting, yes, about how AI can driven, yeah, the natural product to our health care system. Yeah. Okay. State your name and your question.
Thank you for the opportunity, Mr. Raymond. Before I would like to introduce myself. My name is Safira Ramdania from FFUP and Pancasila University. So before you tell us that AI can be used as discovering and formulation in the industry, of course. because AI can predict molecular properties very efficiently, but experimental validation remains essential, like in vivo formulation. In your experience, what are the biggest limitation of relying on AI predictions in drug discovery and formulation? Thank you.
The biggest limitation about the AI.
The limitation is mostly not in the AI.
Huh.
The limitation is the user of the AI.
Oh,
And because AI is only tool, yeah. So the prompt in which you put into the AI will determine what kind of output that you get from the AI. So limitations is in our head actually. So that's why I ask people, if you want to use AI, practice putting prompts and see what it turns out. Because I never trust whatever's being turned out by the AIs. I always research again, I always ask again, and I change. For example, you use ChatGPT. I ask Cloud, I ask Gemini, I ask the Copilot, for example. Yeah? So I always challenge them. And the more you do that, your brain will cater to whatever's being turned out and the result of the AI. Never trust AI because it does. Now it's good that they have the disclaimer underneath. The AI does not mean that if you use AI, you will have the best argument, the best decision, the best. No, you always have to challenge. So that's why I use at least 4 or 5 different AI, the agentic as well as the algorithm that we use. That's a challenge. Secondly, I'm not finished. Sorry. When you use AI in industry or maybe in a university like this, the system, because you don't use it as a technician, but the whole thing, if you want to automate the whole thing, the whole organization must be able to cultivate AI, meaning the system of the organization will have to be revamped. It has to be rebooted, so that it will allow that to get the change of the result of the AI as much as we can. As a researcher, you can use AI, but as individual, but in organization, probably what you depend upon and you get the result from AI will determine and will influence the next department. So that's why the whole thing has to be, for example, formulation, going to analytical, going to quality control, going to production. This has to be, the whole organization must be able to think like AI, and the same way thinking like AI. Not like a formulation, just because it's only my work, I don't care about the analytical, I don't care about things. Oh, another one. Maybe in the Fakultas Farmasi, Prof. Dian, if you can have a class on packaging development. That is very important. Packaging development does not belong to seni rupa nowadays, not in the media. Yeah? No, pharmacists has to be able to do that. The reason our packaging is more complicated and we have to do leaching studies. How much interaction between our drugs and the packaging. That requires leaching studies, and that becomes more difficult because now we have injectable, which a lot of leaching in the top of the injectable, sometimes it's because of the rubber, whatever the material. We have to do studies on those. So packaging development is an area of pharmaceutical industry which we need a lot of expertise on. Going back to you. So the AI from the pharmaceutical formulation, from the analytical, coming the maybe going to the quality, going to the production, the packaging development, until this QC release. This has to be AI. It has to be talking to one another. Not one AI here does not talk to the other. No. And then going back on top of that, regulation. Yeah, some of you as a regular in the pharmaceutical industry, you can work as a regulatory affairs, which has to go to POM, and if you export HSA in Singapore, whatever. Yeah. So.
okay. The AI is a way of our life, and then we have to connect to all of us to can develop or use AI well. Okay, thank you. Mr. Raymond. Yeah. Anything? Any question so far? From the Philippines? Dr. Iqbal. The second question from Dr. Iqbal.
Yes.
Okay.
Okay, Prof. Raymond, thank you for your exciting explanations. I have a question in the point of view of a researcher who work in the pharmaceutical industry, for example. And you have to choose which is bioactive fraction or bioactive compound. Which one is better? So we have to consider pharmacological aspect. We have to consider economical aspects to product to be product to be productive the product. Which one is better, bioactive fraction or bioactive compound?
Okay, which one is better, bioactive compound or bioactive fraction?
Very good question, Dr. Iqbal. Yeah, I've been thinking about this since 20 years ago, actually. Now, unfortunately, our friends in Badan POM, sometimes I feel, only limited by this wall, yeah. Sometimes it's hard to get number registration with
Registration.
compound. But it's our ideal. You isolated a compound, you study pharmacological, as if like you have a new chemical entity. But our friend Badan POM will not readily recognize your, people ask TD, tambahan data. Oh, it will take probably 10 years to get your proof. So the next simple thing is bioactive fraction. Yeah. But if you find a bioactive compound, you can register the supplement outside Indonesia.
Okay.
In Singapore, in the Philippines, you can readily register, but not in Indonesia. But you cannot, if you find something in Indonesia, you cannot readily export. Why? Because they ask for certificate of pharmaceutical product, CPP, meaning it's a certificate in which showing that this invention has been sold and has been sold in Indonesia. Yeah, that's a requirement. So, tough one. Bioactive fraction is the next. It's closest to bioactive compound.
Okay, we know that.
Okay, thank you, Prof. Raymond. So there is reason behind why you insist in bioactive fraction than bioactive compound.
Yeah,
Thank you, Prof.
Absolutely. Extract is not specific enough for me. This is the middle. Bioactive compound, if our Badan POM has changed the way they think, I would go with that. But not now, as we speak.
Okay, thank you, Prof. Raymond.
Okay, about the bioactive fraction with mixing all of the simplicia to one fraction.
Okay. The question. This is another good question. Yeah. Do we use mixing of simplicia? Yes.
Yeah.
I like to mix the simplicia and not a fraction, one simplicia, another fraction simplicia, and then at the end mix. No, I don't like that.
Okay.
Yeah. I'd like to mix the simplicia to get the fraction, but bear in mind, do not mix more than two type of simplicia. The reason being, it's hard to get the fractionation of all the compound. Just remember, one simplicia can have probably about 300 compounds at the most. Yeah? Another simplicia maybe 50. Then you have 350, and then you get only like 5 small one, right? From that bioactive fraction. So it's hard to get it. If you mix like jamu, you have 10 simplicia. That becomes again interaction.
Yeah. There money interaction with maybe 2 and 3 simplicia.
Exactly.
If we mix. It's
I
okay.
don't want to mix more than.
Okay. About all your product, Mr. We know that DLBS 3. Okay. In Lasin and the many kind of DLBS product. We know that all the products come from fraction.
Yes.
Yeah,
Absolutely.
fraction. Okay.
So, for example, my friends here, from Valeria macrocarpa, we have 5 products already. One for cancer, another one for the liver, another one anti-inflammation, another one good for dysmenorrhea, for endometriosis, Another one, I think, is for something else, but 5 fractions.
Five
Just
fraction.
from kayu manis, which is cinnamon
Cinnamon.
burmani.
Cinnamon
By the way,
burmani.
the best kayu manis pharmacologically is from kerinci.
Oh.
People says, oh, Sri Lanka has kayu manis. India, no. I collect all the from even South America has Kalimantan has not good pharmacologically. The best is kerinci. We're so lucky we have the best cinnamon burmani. Not
From
cinnamon
kerinci.
Lanika, not cinnamon yang lain. No. Cinnamon burmani is the best pharmacologically. You don't have to repeat already because I've done it. So if you want to use cinnamon, just use burmani.
Okay.
If you want to open a good bakery, then you use Selanika, which is coming from Ceylon, because the smell is good. Even when you cook it, you want to eat it raw because it smells good.
Smell good.
Cinnamaldehyde.
Cinnamaldehyde. From this.
Produce fragrance of cinnamon.
Okay.
So, and in the mall, there's Cinnabon, whatever. So
Cinnamon.
use cinnamon from Ceylon, from Sri Lanka.
Pricey.
Yeah.
Pricey. Okay. There is some a lot of question. Any questions so far? Okay. Can you stand up and state your name? Oh. Yeah.
Good afternoon, Professor Raymond. My name is Alfred
Good afternoon.
Raymond from
Yeah.
Pancasila University. Thank you for your insightful presentation. I have one question. How can universities and students contribute to protecting Indonesia's biodiversity while also utilizing it for drug discovery? What do you think is the most effective way to balance biodiversity conservation with pharmaceutical research and innovation? Thank you.
How?
Yeah. Repeat very slowly, yeah.
Just slowly, please, slowly. How?
Wait, how can universities and students contribute to protecting Indonesia's biodiversity while also utilizing it
Protecting biodiversity.
How do you think is the most effective way to balance biodiversity conservation with pharmaceutical research and innovation?
Okay, so your question is protecting biodiversity.
Yeah.
Oh, several ways. Number one, if you can prevent kebakaran.
In Kalimantan.
Fire in the jungle, Malaysia,
Kalimantan.
in Papua. Because number one contributor of biodiversity, the destroying of biodiversity is kebakaran, is fire. Yeah. Number two, you talk to the company like Sinar Mas. All they do is actually destroying all the all the forest. That's another protecting biodiversity. Number three, let's take care of all the plants. If you go to Bali, if you go to anywhere, you go snorkeling, do not destroy the rock. Do not destroy what you call it's just ecosystem with that underneath the sea. Many people like to destroy, like for example, Raja Ampat's already somewhat destroyed. That's protecting biodiversity. So it's because your generation will have a much less biodiversity what we did, but what we had before. So you have to have again, this requires legislation that people do not destroy readily the biodiversity. And second of your question is, as a student, how can you provide, what was the question again? Number two, even you forget what the question is.
Yes, what is the most effective way
The most effective way
to balance biodiversity conservation with pharmaceutical research and innovation?
to balance.
Yeah. So the one that the examples that I show you, I do not use too many plants.
Oh,
That's one way to balance. The reason being, if you use too many plants, how are you going to source them? And it's not easy to convince our farmers to plant the biodiversity anymore. Why? Because some farmers in the past, they're so hopeful that some industry will buy their plantation, the result of the farming. It turns out, no. Some of our extract is still being imported to this country, like it or not. Some of the jamu that you take, whether it's jamu whatever, jamu Borobudur, jamu whatever, itu the import. We still import them. So that's why if you have a good solid research, do not use too many plants. Use only selected plants, but you make bioactive fraction derivatives. That's why you still contributing to the preservation of all these raw materials, of all the biodiversity. So don't use too many plants. Use one or two at the most, and you make bioactive fraction.
Okay, don't use many plants. Just 1 and 2 plants. Yeah, to maybe make some medicine. Yeah.
Yeah, I just give you an example. Jamu for dysmenorrhea. You know what dysmenorrhea is? When you
Okay.
have menstruation, you get painful.
Menstruation.
Yeah, you find jamu for dysmenorrhea into your Google. At least it has different 8 ingredients,
Okay.
7 or 8 ingredients. Why? In the old days, we don't have the technology. We don't have the bioinformatics. We don't have AI. We don't have anything. So what I did is I found a good bioactive fraction which will mimic the 8 different. So just use one bioactive fraction for malaria macrocarpa. It actually mimics the whole scheme of the 8 different jamu, jamu with 8 different ingredients. That you do not need too many ingredients, because otherwise the cost of making one ingredient is high, especially if you put 8, even higher.
Interesting.
Yeah, and it's going not good for the body.
Okay.
Too much. Yeah.
Maybe the last question from the University Philippines, maybe? The last question.
Yeah, it's finished.
Can you state your name and ask the question?
Good morning. I am John Ray Valenzuela from the Philippines, one of the delegates from Trinity University of Asia. And my question is in regards to the financial aspect of your presentation. So I'd like, how strong is the financial support this instructions, considering phytochemical content is based on so many environmental factors, as well as preparation techniques, knowing that these methods and these techniques are very expensive, and it takes a really long time to produce the results that we want to achieve.
Yeah, thank you very much. Salamat for your questions. Thank you so much for your questions. Yeah, that's a very good question. Yeah. How can we tone down the what we call the COGS, cost of goods sold, production price? Yeah, that's what your question. Yeah. Now, number one, this is a very good question, because as a researcher, you do not think only for the technical formulation,
Okay.
extraction. No. How can I design a product which will meet my COGS? For example, I want to sell it, like for example, how much? Yeah, in peso, maybe. How much is metformin in the Philippines? Do you know what the price of metformin is in peso?
How much in peso?
13 to 15. That's a branded. Generic. Yeah, okay. 13 to 15. So for example, the generic is 30 or 50. Yeah, maybe. Yeah, 113 is generic. If you make it the originator, maybe 10 times as much. Yeah. So you want the COGS about maybe 15 at the most. In order to do that, then you have to find a good bioactive fraction which allow me as a researcher to get the same amount of efficacy into my bioactive fraction. And the reason why doing that is, in the future, my product will have to compete in the government sector to the generic. So that's why you have to cut down. You have to increase the yield and cut down the amount of COGS. So that's why, for example, even my thinking is, even if you go to 20 pesos, still okay. People, if it's a new invention, government still full help, yeah, we'll still have to, reimburse. And we've done this, we've done the exercise with the Ayala group in the Philippines, in Manila, and we found that, okay, it is probably about 10 to 15 or even 20% higher than a regular generic product. In order to do that, that's why you have to do quite a bit of knowing how to find a new formulation, yeah, and new analytical, and make sure that the cost of production will not increase as high as an originator product. Like, for example, a product called empagliflozin, maybe it's around, I don't know, maybe 100 peso, maybe. Empagliflozin is a brand new drug, maybe about 5 years. Our cost of target maybe only 20 peso instead of 300 for diabetes drug.
Okay.
That answers.
We talk about the COGS. Maybe you know that the natural product, especially in Indonesia, may be more pricey. So we have to learn how to make our product COGS is acceptable to our people in Indonesia. Okay, thank you very much for all of you participants, and thank you very much for Professor Raymond.
Can I have a questions, like last questions, Pak Zainul?
Okay.
Okay, because,
For you,
yeah, I'm very excited.
Mr.
to
Paradiba,
you.
Mrs. Paradiba. Ask you.
Yeah,
Okay.
because Pak Iqbal have opportunity, then I might
Okay.
have also. I will use this opportunity to officially introduce myself to you, Prof. Raymond. My name is Faradiba, and I'm here also a lecturer in Faculty of Pharmacy, but we kind of a different area of knowledge. I'm in policy and management of
Policy.
pharmacy. So following up the question from the last question about COGS, from your presentation, I can conclude few points. One is to advance and to moving forward with traditional medicine. We have, a very basic challenges, like you said, human resource, and then health financing of the government that must set the price or the ceiling price very low because we have a limited budget, especially for now, because the political window, etc. and etc. And my question is, but also you pointed out in your presentation, AI may help us as a tool that maybe, I don't know, I have to confirm with you, maybe, is it possible to use AI in the process of research and development of traditional medicine to possibly reduce the price of the product? Is that what you are saying, or I misunderstanding here? Thank you, Professor.
Thank you. Good question.
Paradiba.
Okay,
Mrs.
thank you.
Paradiba.
Okay, good question, Mr. Faradiba. Now, yes, we use AI, obviously. Yeah. Now, in your field, we do quite a bit of pharmacoeconomic studies. Yeah, with the HTA, health technology analysis. We found out that the most important thing is not COGS with the pharmacoeconomics. It's actually, does it reduce the whole total cost of therapy? So when you compare that, for example, a diabetes drug from the biodiversity to metformin being it's only like 50 rupiah, 100 rupiah, then your cost should be, sometimes it's a little bit higher than metformin, but if it reduces the cost associated with the treatment, direct and indirect costs, it has good cost-benefit analysis, CPA analysis, or even cost-effectiveness analysis, whatever you're doing, cost-utility analysis, whatever you're doing, it's still being acknowledged as a good pharmacoeconomic product. So when we do that, we've argued before the Ministry of Health, please don't take a look at only the cost of the product. Take a look at the cost of therapy. Because actually, if you have diabetes, not only lowering the blood glucose level, but also you can prevent micro complication, stroke, for example, the myocardial infarction, even DVT, many things
then you can put that into account. Obviously, the direct and indirect cost put into the equation too. Then you can convincingly say, yeah, maybe the COGS is a bit higher, but I can save much more because I use this product coming from the natural product, because the cost of therapy is much lower than if I've used only metformin or glyburide or whatever, glycoside and so on. Yeah, hopefully that answers it.
Thanks. Okay, thank you very much for Professor Raymond for the enlightening answer. Good macroeconomy product. Oh, this is interesting. So we can see all costs, yeah? Okay. Distinguished guests, ladies and gentlemen, before we conclude the first session, allow me to summarize the key takeaway from Professor Raymond. The first, all shift the pharmaceutical R&D mechanism for linear trial error process into predictive data-driven system. Tasks like target discovery, molecular design, and clinical testing now heavily utilized in silico simulation to dramatically reduce high R&D costs and clinical test failures. Phytopharmaca cannot move simply as standalone product. It must move as the system. So we incorporating phytopharmaca into national healthcare insurance, maybe BPJS. This integration will increase the production volume, yeah, and so on. How the crucial is to integrate natural product system. Okay, in conclusion, bridging advanced data science like AI with clinically proven natural biodiversity is indeed the blueprint for the future of global pharmacy and national resilience. Please give another round of applause for extraordinary speaker, Professor Dr. Raymond Chandrawinata. As your moderator, I apologize if any mistake. Thank you for your join and active participation, and I will now hand to the MC. Thank you very much, Mr. Thank you for all of you.
Thank you so much, Professor Raymond, for your insightful materials. We also sincerely thank our moderator, Mr. Zainur, for leading today's session. But please stay in the stage because we will present the certificate, and we would like to invite Mr. Andre Prasetyo to present the certificate. From the documentation team, please take a picture in 3, 2, 1. Once again. 3, 2, 1. Okay, thank you so much.
For our second material, we will be given by Dr. Sandi Kelintang, Master of Biomedic, President Director di Pharma Adiluhung. To begin this session, we would like to invite our moderator, Apotheker Faradiba, Master of Pharmacy, to the stage.
Cek. Udah ya? Cek. Okay. Yeah. Thank you. Thank you for the kind introduction, Master of Ceremony. Good morning, everyone. Is it me or are you feel happy here? Yes, because we have the brightest scientists in Indonesia. One is Prof. Raymond, as you know, and as
previous session that he highlighted many points about how we can improve our ecosystem in traditional medicine, etc. And now I'm feeling honored to be moderator, our second speaker. He is also the brightest in Indonesian scientists, just like Prof. Raymond said that Dr. Sandi Klintang is one of, let me start from the beginning, okay? Distinguished speaker, respected faculty member, and dear participant from Indonesia and the Philippines, good morning, and we have a very delightful session with you today. It is a great pleasure to welcome International Summer Course in Pharmacy organized by Faculty of Pharmacy, Universitas Pancasila. I hope everyone's enjoyed today's inspiring session and gaining valuable insight from our international discussion on the future of pharmacy. My name is Faradida and I'm honored to serve as the moderator of the session. It is now my privilege to introduce our second speaker for this morning. Please join me in welcoming Dr. Sandi Klintang, Master of Biomedicine, President Director of Pharma Adiluhung. We will deliver a presentation entitled Bridging Botanical Wisdom and Regenerative Medicine, the Potential of Plant Extract in Cell Therapy. Before we begin, allow me to briefly introduce our distinguished speaker. Dr. Sandi Klintang, Master of Biomedicine, is a physician, scientist, and industry leader with more than 2 decades of experience spanning biotechnology, regenerative medicine, and the pharmaceutical industry. He currently serves as a President Director of Pharma Adiluhung and has held several prominent leadership positions, including Deputy Director of Stem Cell and Cancer Institutes under Kalbe and President Director of Kalgen DNA. Throughout his career, Dr. Sandi has been at the front of advancing stem cell and cell-based therapy in Indonesia. He has actively contributed to the National Research and Innovation Initiative as a reviewer for the Ministry of Research and Technology, BRIN, and LPDP, while also participating in the development of policies and quality standards for advanced therapy medical products in Indonesia. His expertise has been recognized internationally, of course, through invited presentation in Singapore, South Korea, Taiwan, China, Japan, and United States, where he has shared insight on stem cell, regenerative medicine, and the commercialization of cell therapy. And he was honored with the Best Speaker Award at the Annual Congress on Cell Science and Regenerative Medicine. Beyond his scientific achievement, Dr. Sandi is widely respected for his commitment to bridging academia, industry, and clinical practice, transforming scientific discoveries into innovation that has the potential to improve patients' life. Without any ado, because he is very phenomenal, so the CV might take longer than that, but I just give a brief curriculum vitae of his phenomenal hard work. Ladies and gentlemen, please join me a warm applause to welcome Dr. Sandi. Cek dulu Pak, cek dulu. Harusnya udah. Tinggiin lagi kali.
Hello, test. Okay, good. Okay, good morning, everyone. It's an honor to be here at the third International Summer Course in Farmasi, Universitas Pancasila. And today we will explore a fascinating intersection, how ancient botanical wisdom can inform the future of regenerative medicine. And I look forward to sharing our findings on the potential of plant extract in cell therapy. Okay. And this slide starts with a simple image.
growing a rice field. For strong seedlings, you need sunlight, waters, and fertile soil. Yeah, I think it's everyone is agree for this. Our new cells are no different. They need good oxygen, nutrition, and healthy tissue. Yeah. For strong seedlings, you need sunlight, also water and fertile soil. Our new cells are no different. Yeah. And so where do plant extract come in? They like natural fertilizers. They reduce inflammations, improve tissue health, and prepare the soil so new cells can thrive. And the next slide. Today we will look at 4 main types of stem cells, organized by their source and potential. First, embryonic stem cell. Come from the early stage embryos. They are pluripotent, meaning they can form any cell type in the body, and they are highly versatile, but come with ethical concern. And the second is adult stem cell. Are found in tissue like bone marrow, fat, and they are multipotent, so they are limited to specific cell types. For example, bone, fat, and cartilage. They support natural repair and have no major ethical issue. And third is a perinatal stem cell, are collected from birth, related tissue like umbilical cord, blood, and lining. And their potency varies, but they are often multipotent. They are easy to obtain and raise fewer ethical questions. And finally, induced pluripotent stem cell, or iPSC. And this product or stem cell we have in our facility are created in the lab by reprogramming adult cells, like skin cells, and back to a pluripotent state. This allows for patient-specific models and is valuable for disease research and drug testing. Based on the ICT, critical criteria for defining MSCs. And to define MSCs, ICT established 3 minimum criteria in 2006. First, they must adhere to specific in standard culture. And second, they need a specific marker profile, positive for CD73, CD90, CD105, and negative for hematotic markers like a CD45 and SLDR. Third, they must show lineage differentiation into bone, fat, and cartilage, because they come from the mesodermal or multipotent. And finally, they must have robust self-renewal, allowing large-scale expansions. These 4 points remain the gold standard for MSC identity. And after birth, where the MSC are found? MSC are found in every perivascular niche surrounding blood vessels throughout all our body. Their 2 main functions are protection and repair. First, they suppress inflammation and calm the immune response. And the second, they promote tissue regeneration and repair damage. So, MSC act as a built-in defense and healing system located right next to our blood supply. If you look, the diagram shows the 2 main ways MSC work. First, they moderate inflammation. They reduce inflammation, shift macrophages to a healing state, and decrease cell death and cytotoxicity. And the second, they promote repair. They release growth factors, cytokines, and exosomes, which enhance angiogenesis, support tissue scaffolding, and regenerate parenchymal cells like adipocytes and myocytes. In short, MSC don't just replace cells, they create a healing environment. And this slide introduces the secretome. Everything stem cell secret. MSC are master managers of this secretome. They release signaling molecules like growth factors and cytokines to communicate, regulate, and maintain homeostasis. Importantly, the secretome is dynamic. It changes with environment condition, nutrient, oxygen level, and culture conditions, and also during differentiation into specialized cell types. This is why understanding and manipulating the secretome holds great promise for regenerative medicine, with potential therapy for neurological, cardiovascular, and other diseases. In short, stem cell heal not just by becoming new cells, but by managing what they secrete. And we look now shifting gears. This paper review antioxidant phytochemicals from natural sources like berries, pomegranates, medicinal plants, and more. Key compounds include resveratrol, curcumin, quercetin, and lycopene. They work by reducing oxidative stress and reactive species. The health benefits are broad, improving cardiovascular function, lowering blood glucose for diabetes, protecting against neurodegeneration like Alzheimer, and preventing DNA damage in inflammatory bowel disease. In summary, these phytochemicals act on multiple targets. Future research should focus on their mechanisms and potential adverse effects. And the botanical arsenal in regenerative medicine, we see the building on that review of antioxidant phytochemical. This slide takes us a step further from prevention to regeneration. And while the previous paper focused on managing oxidative stress in chronic diseases, here we see a more targeted botanical arsenal. Its plant isn't just an antioxidant, it has a specific cellular job. Turmeric fight inflammation at the pathway level. Aloe drives cellular repair, and ashwagandha actually modulates the stress response. This moves us from reducing damage to actively repairing and adapting at the cellular level. And the next slide, so we move from chronic disease prevention to targeted cellular repair. Now, this slide shows us how these botanicals actually interface with the cell therapy. The challenge has always been keeping cells alive and avoiding immune rejection. That's the bottleneck. But nature offers a solution. These plant compounds provide biomimetic support. They are like pre-optimized chemical scaffold that help cells survive and integrate. The real breakthrough is synergy. Phytochemicals don't just protect cells. They actively enhance them and stimulating stem cell differentiation, modulating immunity, and accelerating functional tissue repair. This is where preventions meet regeneration. So, we establish that phytochemicals and cells work synergistically. But how do we actually apply this in practice? This slide breaks it down into two distinct pillars. The first is the manufacturing boost. This happens in the lab before the patient ever receives the cell. Here, phytochemicals act as signaling molecules to increase MSC proliferation and guide them towards specific lineage like bone and or cartilage. The second pillar is the clinical shield. This happens inside the patient. Once transplanted MSCs face a hostile, inflamed environment, and phytochemicals protect them from cell death, reduce oxidative stress, and prime them to secrete healing factors. So, it's a dual strategy. Prepare the cell before and protect them after. As we talk about the strategy, preparing MSC in the lab and protecting them in the clinic, but what are the actual molecules driving this? This slide gives us the roster. On the left, a wide array of phytochemicals from curcumin and resveratrol to genistein and nerigin. On the right, key vitamin like A, C, D, and A. And in the center, the shared target is stem cell. The arrows tell the story. These compounds actively promote both proliferation and differentiation. So this isn't just a list, it's a toolkit. The same molecules that boost cell numbers also guide them toward becoming the specific cell types we need for regeneration. And we've seen the molecule that regulate stem cell. But knowing which compounds to use is only half the battle. This slide addresses the how. How do we actually extract these bioactive compounds to maximize stem cell quality? The top panel shows the problem with standard extraction. Low viability, cell stress, inconsistent composition. The bottom panel shows the solution, an optimized extract plan that maintains cell integrity, minimize stress, and ensure consistent quality. The result, high quality stem cell and a superior secretome with increased therapeutic potential. So, this is really about manufacturing excellence, bringing the gap between knowing which molecules work and actually delivering them effectively. And this dashboard summarizes 4 key mechanisms by which natural products reprogramming the cellular environment. First, they suppress inflammation by inhibiting NF-kappa B, MAPK, and interleukin-6, breaking the degradation loop. And second, they combat oxidative stress by scavenging ROS and nitric oxide, protecting the extracellular matrix. And third, the active antioxidant defense through the NF2-SO1 pathway, enhancing MSC resilience. And fourth, they modulate cellular lifespan via SIRT-T1 and reducing senescence and improving mitochondrial dynamics. So we built the case for optimized stem cell manufacturing. High quality cells, superior secretome, consistent bioactivity, but why does this matter clinically? This slide gives us the answer. Osteoarthritis, for example, we see the progression here from intact cartilage to early degradation, then moderate changes with inflammation and osteophyte, and finally severe joint destruction. And when we look at current treatment on the right, the picture is sobering. NSAID offer only temporary relief, injection have variable efficacy, surgery carries risk and finite lifespan. None of these stop disease progression. And this is the clinical gap, and it's precisely where our optimized stem cell and secretome therapy could fit. And if we see the slide, at the bottom is stem cell therapy. This is where our optimized phytochemical enhanced MSC fit in. They offer the potential to not just reduce pain, but to modulate inflammation and promote tissue regeneration. So we are not starting from scratch. We are building on this emerging foundation, but with a more refined phytochemical boosted approach. We are positioned stem cell therapy within the osteoarthritis treatment landscape. Now let's look under the hood at how bioactive compounds actually enhance MSC performance for cartilage repair. The input is straightforward. Curcumin, resveratrol, quercetin, and EGCG is a biologically active catechin in green tea. Its compounds we have discussed earlier. They work through 2 key mechanisms. First, they mitigate oxidative stress at the injury site, reducing the high ROS environment that would otherwise damage cells. And the second, they promote chondrogenic differentiation, guiding MSC to become healthy chondrocytes that produce collagen 2 and agrecan, the building blocks of cartilage. The result, enhanced MSC performance from expansion and homing to actual cartilage formation, repairing defect and restoring joint function. And the next slide, I want to introduce this establish the mechanism how phytochemical enhance MSC performance and drive cartilage repair, but does this translate to patient? This slide introduces clinical evidence, and the study published in the Journal of Clinical Medicine investigated umbilical cord MSC secretome, not the cell themselves, but the paracrine factors they secrete in knee osteoarthritis. The key finding, it improved clinical outcomes and changed biomarkers. This is important because it validates the concept we've been building, that the MSC secretome, particularly when optimized, has therapeutic potential, and as we discussed, phytochemical can enhance that secretome, making it even more potent. And we focus heavily on osteoarthritis, cartilage repair, MSC secretome clinical outcomes, but the potential of phytochemical enhanced stem cell therapy extend far beyond the joint. This slide applies the same concept to neurological disorders. Flavonoids, plant-derived compounds with potent antioxidant, anti-inflammatory, and neuroprotective properties, work synergistically with stem cell. The mechanism is consistent. Flavonoids improve the microenvironment, enhancing stem cell survival, proliferation, and integration. The targets are some of the most challenging neurological conditions is Parkinson's disease. So the strategy we have developed for cartilage repair is actually a blueprint applicable across multiple degenerative conditions. And the next slide, if we look, is the video of the Parkinson before and after give the stem cell and combine the phytochemical. And the next video, and after get the stem cell only in 3 weeks, actually the patient already has a Parkinson around 8 years, and then he already got the standard treatment of Parkinson, but the progression, this is a neurodegenerative disease, and after get the stem cell combine the phytochemical, the result is very good and it's very promising because only 3 weeks the patient can the normal person. And to summarize, we shown that phytochemicals enhance every stage of stem cell therapy, from expansions to differentiation to survival. We have validated this in cartilage repair, demonstrated clinical promise in osteoarthritis, and extended the concept to neurological degenerative. But we are not finished. The research priorities are clear. Optimize compounds in vitro, test in clinical model, and translate to the clinical. And we can do it alone. We invite collaboration from researchers, scientists, and clinicians. Let's drive innovations together. Thank you very much for your attention.
Warm applause. Check. Check. Okay. Well, applause once again for Dr. Chintan. Thank you, Doctor. Thank you very much for a thought-provoking presentation, and I think we all agree that you have a very remarkable work. And I just note a few points from your presentation. It's kind of a lot, but I just have a brief. I think we have a very interesting discussion today with two best scientists in Indonesia, because with Prof. Rayman, we talk about the challenges, the opportunities of Indonesia, and also the position of Indonesia. And he gave us many examples on how we can advance our knowledge and effort to traditional medicine. And, it's kind of macro ecosystem with Prof. Raymond, and then we have Dr. Sandi Klintang for the micro environment, like a very theoretical approach that he has been presented, as you see. So I have a few points from your presentation. First, you pointed out about stem cell, and there are also the criteria, the source of the stem cell. And we have medicinal signaling cells, okay, and the role of phytochemicals to maximize that work to regulate the stem cell to make it better regenerative medicine. And then you also pointed out about extract plant, which is a very manufacturing excellency, if I may say, because the place where Dr. Sandi work is one of the best in the nation have. Like I said, the Farmasi, the Dukung, and then DLBS, you name it, all the Talbot Group's company have a very excellency in manufacturing. And then you also give us an example about osteoarthritis, which having a clinical gap, and then stem cell therapy filling the gap. And the last but not least, I have the role of flavonoid for Parkinson's disease. So we have two examples, osteoarthritis and then Parkinson's disease. So we have a lot of, I think we have a lot of questions from all of you guys. Hello, yes, I know. Okay, so I think you have to prepare your question because we will move on the Q&A session. Dr. Sandi, do you want to answer one by one or we collect one by one? Okay, so one by one, please, for the Philippines, state your name and institution.
Good morning. I am Francesca Lim from the Philippines, a delegate from the Trinity University of Asia. In your opinion, how do gene editing technologies such as CRISPR-Cas9 complement stem cell research in developing future therapies for neurological diseases such as Huntington disease, Alzheimer's, and Parkinson's? Thank you.
Okay, it's very good question, yeah, about the CRISPR-Cas9. I think this technology is quite new and it's very expensive. And actually, in US, I think there's a few diseases already using the stem cell with the CRISPR-Cas9, like for thalassemia or even from HIV, as I know. This is a part of the gene editing. And if we look the stem cell criteria therapy, the stem cell therapy criteria based on the international criteria is a part of the ATMP, Advanced Therapy Medicinal Products. There is a four criteria. First is stem cell, and the second is the gene therapy. Third is scaffold, and then fourth is and third is scaffold, and fourth is tissue engineering. And come back to your question is MSc is CRISPR Cas9 because it's very expensive. Now in Indonesia, as I mentioned, we have the iPSC, induced pluripotent stem cell. It is from Simayan Manata. His guys already got the Nobel Prize in 2012, and he reprogramming the adult stem cell or adult cell, like your skin cell, become to the like embryonic stem cell, like a pluripotent stem cell. And we have now, and we can, I think this is very important to do the research, because the iPSC, we can differentiate this stem cell to any cell, and we can how the efficacy of the phytochemicals to to any cell in our body. So we don't need the animal cell, but we can using the iPSC is the human cell. It's the real cell in the research, not the animal cell. I think we can more nearest about the biological cell in the human life rather than the animal cell we can use. Thank you.
Is that enough? Okay. Second questions, please. Okay.
Good morning, Dr. Lintang. Thank you for the presentation. Let me introduce myself. My name is Gaya Kusuma from University of Pancasila. Let me ask a question. Indonesia is known for its rich biodiversity. Which Indonesian medicinal plants do you believe hold the most promise for future application in cell therapy? Thank you.
Yes, I'm very believe about this because my experience to using the stem cell for any person now maybe around more than 10,000 people already using the stem cell, especially when the COVID-19, so many patients using the stem cell, especially for the critical or severe and critical COVID-19 using the stem cell and the secretome. And I very believe we combine the stem cell with the phytochemical. But the important thing, what kind of phytochemical we use? Well, because I believe the phytochemicals is a targeted cell. Like for osteoarthritis, is the kind of special phytochemicals. For neurodegenerative, like as the unique phytochemicals we can use. So the combine of the stem cell treatment with the phytochemical, I think is more make it the stem cell more potent. And the important thing, we can do the research in the in vitro, using the stem cell co-culture with the phytochemical. So what the best phytochemical extraction using to see the increase the quality of the stem cell, like proliferation or differentiation, and we can get the best of phytochemical extraction. I think that. Thank you.
Yeah. So following, is there any questions? Okay. Okay. So yeah, maybe from the front, from the Philippines.
First of all, thank you for the opportunities. Good morning, Dr. Sandy and Ms. Faradiba. So I would like to ask a question regarding the application of stem cells, especially in the National Health Insurance system, considering I just think that it is relatively more expensive. So how do you think, does it has the potential to become the first line therapeutic options compared to the conventional chemical drugs in accordance to the National Health Insurance system?
Okay, thank you. We know the National Health Insurance is a very high burden about the degenerative disease, and so many trillion rupiah, they debt to pay to the hospital. And I think the important thing is lifestyle. And as I mentioned in my slide, there is a stem cell in our body, in all of the perivascular blood. And I think my stem cell and your stem cell, I think the best in your stem cell, because your age is still young rather than me. I am now more nearest the 60 years old. And 60 years old. And you maybe around 20 or less than 30 maybe. And the important thing, how to maintain your stem cell. Yeah. So if we decrease the national health insurance burden, we should educate the people or society how to good lifestyle. And also maybe the phytochemicals can we propose to the government to maintain the quality of endogenous stem cell. Since when? Since young people. Since young, like you. Not, yeah, maybe I'm too late. But I still maintain my stem cells also. But with what? Maybe I still maintain my stem cell with the phytochemical also. Yeah, food supplement I intake. And also I routinely secretome to my body. Because I'm too late maybe, yeah. But from the young age, maybe still just need a phytochemical. And if you do the discipline of this, maybe the next generation, the degenerative diseases quite decrease. Because and the important things from the food, yeah, because so many sugar in the food now. Yeah, because be careful. Like sorry to say the name is Starbucks, for example. It's very many sugar, not rather than coffee, I think. Yeah, so be careful about this food or drink you intake. Yeah. Thank you.
Yeah, I think it's really interesting point, Dr. Sandy, you mentioned about lifestyle because you know the new generation like me and the younger is having shifting lifestyle. Like in other side we have like coffee or tea with many sugar, like you mentioned. But in other side, we also having increasing awareness about physical activity. I think that's also a good point. So if I may conclude that sugar is our enemy. Yeah, sugar is our enemy for some context or for general context. And what Dr. Sandy said also, we have to maintain our stem cells because it's related to maybe quality of life and our preventive effort for ourselves to prevent any disease coming in the future. Okay, that's really good point. Thank you, Dr. Sandy. But I just want to following up the question about what you said before. In the context of universal health coverage or BPJS, because it's always about limit budget from the government. Where are we? From your perspective, where are we now? Are we on the right track to, let's say, making the stem cell therapy becoming our primary therapy, or we still far from that? Where are we?
Yeah, we know the stem cell or secretome is very expensive. Like stem cell, around 250 million rupiah per one injection. It's like one car, Avanza. And for secretome, around 2 or 3 million rupiah per injection. And the patient needs routinely at least one a week. Yeah, one a week, 2 or 3 millions. And in a month, it's around 12 million. It's very expensive. So, but it's okay. The stem cell or secretome still in using in the high class segment. But it is the important thing is for the no option disease or a rare disease. The no option disease, there is no chemical compounds or anything treatment can in the patient. And then finally we can using the stem cell like a Parkinson,
Yeah.
Alzheimer, stroke. Yeah. But for the national coverage, I think because we know the stem cell now, the knowledge of stem cell, and then we know our bodies, there's a stem cell from the after we birth, so we can propose the national health officer or the minister of health or government. The important thing, educate the
People.
Society or people how to make it the maintain of our stem cell endogenous quality by quality of life. Maybe we can using the phytochemical. After we can research the how to phytochemicals can maintain the quality of stem cell with in vitro, and we can propose to the government eat the phytochemicals. Phytochemicals is very cheap, I think. And we can educate the market. Please eat or maintain your stem cell endogenous with the consumption in the phytochemicals. This for the osteoarthritis, this for the neurodegenerative. And then the important thing is we know the development of the genomic in Indonesia. And the genomic can predict the disease. So we can try how the prediction of our disease based on our genomic. And we can using the phytochemicals targeted with what kind of the disease of our genomic test from the genomic test. Like for example, we have the predictive of the genomic based on genomic test is a neurodegenerative. So since now we can consume the flavonoids to prevent the stem cell for our brain.
Oh wow! Yeah.
Maybe you can research do this.
Yeah, it's really interesting. And when you say about genomics, it's kind of individual therapy, right? A very individual therapy rather than community therapy. Okay. Next questions, please.
Good morning. I'm Takeshi Julian from Trinity University of Asia. So my question is, how can we ethically obtain and use stem cells for future researches and for evidence-based therapy? So basically, what are the ethical considerations of collecting stem cells for research?
Okay. Yeah, for research stem cell now, first because our topic is a natural product, natural plant, we can use the iPSC and derive the iPSC to make the differentiate to any cell, like for the brain cell, for the pancreatic cell, and we can co-culture with the phytochemicals. And what kind of phytochemicals can effective to prevent for diabetic, for the neurodegenerative, after we iPSC to derive to any kind of cell because we have now in Indonesia. First, and the second, for the stem cell now, we in our facility we develop the iPSC, derive the any cell, and after that we take the secret of the any cell, like a secretome, and after the secretome we collect the exosome. The exosome is the very small part of the secretome, is the nanoparticle, and we inject to do the some disease like for the chronic kidney disease, and the result is very good and very promising, and we already got the fundraising from the LPDP around very big fund from the LPDP, around 9 billion rupiah in 3 years to get for the registered product. And so many, like you mentioned, your friend mentioned as a CRISPR-Cas9, anything, but yeah, we should calculate the cost. of course. And then is it applied to the society or our market now? Because if we not apply to the market, it's not used, and then it's very costly for the company.
Okay. So following up your answer, can I say, or can we say that there is no or little ethical issue on the stem cell research?
Yeah, as long as we don't, as long as not use the embryonic stem cell.
Okay, as long as we are not use
Embryonic stem cell.
embryonic stem cell, so we are fine. Is that answer your questions? Okay.
Hey, first of all, thank you for excellent topic of discussion that was present. I'd like to ask a question. So regarding from the topic that was discussed, looking at the potential synergy between phytochemicals and cell therapy, which one is more critical to develop first? The standardization of phytochemicals as supporting raw materials or the optimization of the cell initiation techniques itself?
Is that clear or clear? Not really clear. Can you repeat it?
Looking at the potential synergy between phytochemicals and cell therapy, which one is more critical to develop first? The standardization of phytochemicals as supporting raw materials or the optimization of the cell isolation technique itself?
Okay.
Yeah.
Yeah.
Thank you.
For the efficacy of the stem cell therapy, there I always mention around 3 factors how make it the stem cell therapy can effective and safely. First is the production. The production is very important, and then should be get the GMP certificate from the FDA, like Indonesian FDA. It's a GMP, good manufacturing practice. And because this is important, why? First, like I mentioned, is a 3 character of the stem cell and the impurity, like Prof. Raymond mentioned before.
Yeah.
The impurity is very important because if we manufacture the stem cell, we need the facilities should be very clear. Not used, not forbidden or using antibiotic in the facility. And the second factor is the doctors or clinician, how to administer the stem cell. Like administered from for the intravenously, intrathecally, or intracerebrally. Yeah, it's the important thing. Second, and the third is very important is patient condition. Yeah, so before we using the stem cell to treat the patient, we should fertilize the soil, the body of the patient. So that's why we give the patient some food supplement or maybe phytochemicals before the stem cell, around 1 month before the stem cell. Because, you know, the stem cell is a seed. Seed before we give the seed of the body of patient, we should fertilize the soil or environment of the patient to reduce the inflammation and then to good the microenvironment of the patient before the stem cell can give. And then I think this is very effective. The stem cell make it the stem cell is very potent to the patient result after that.
Is that answer your question?
Yes, thank you.
Thank you.
So much.
About phytochemical standardization?
Yes.
Can you
Phytochemical standardization.
Yes.
Yeah, this is a part of, I think, for part of the Prof. Raymond, the phytochemical standardization. But as I mentioned, we can do the research, in vitro research. What the concentration of phytochemical, phytochemical in co-culture with the stem cell, and what the result of the good concentration is the quality of the stem cell. If the stem cell is make it a high quality after the concentration of the phytochemical, I think this can move forward to research the phytochemical with the certain concentration to do for the scalability for the
Okay.
production, and then give to the animal first, and after that the patient.
Okay. Okay. I think that's really good and interesting point from Dr. Sandy. And more, do we have time, committee? Do we have still? Okay, so we have around 10 or 30 minutes. 15 minutes. Okay, maybe one or two more questions. Yes, please.
Good morning and thank you for the opportunity, Dr. Sandy and Mrs. Faradiba. My name is Natania Felicia Wijaya from University of Pancasila. So you mentioned that stem cells exert many of their therapeutic effects through the secretome. And my question is, do you think plant-derived bioactive compounds could be used to modulate the stem cell secretome to enhance tissue repair and reduce inflammation, potentially making secretome-based therapy more effective than direct stem cell transplantation? Thank you.
Yeah, thank you. It's very good question, I think. So many cells can secrete the secretome, and if we manipulate the stem cell, the content of the secretome is changed. It's very dynamic, and I think it's just like us. If you eat some food and then you smell is different, and like you make it the garlic, you eat the garlic, you sweat smell is like a garlic.
Yeah.
And you eat the pate, your urine is like a smell is pate. It is a secret of our body. So if we manipulate the stem cell with the phytochemical, and the secretome also we can is the dynamic to produce the specific secretome to the target organ. I think it's very interesting to do the research on this project. We can gather together with our facility stem cell and do the research in Universitas Pancasila.
Yeah, I think that's a very good opportunity. Following up that question, Dr. Sandy, what is the smallest thing that the student can do? I mean, for which semester are you guys? 5, 6?
Semester 6.
6.
Yes.
The smallest thing that student can do if they are interested in this kind of knowledge.
Yeah, we can do with animal study or in vitro, maybe we can, or animal study because not a clinical study. For the wound care or for wound care for the animal, we can introduce the animal with the luka bakar, then how to speed up the healing process of the luka is from the burn wound, and we use the secretome, naive secretome, and we can co-culture the stem cell with some phytochemical and introduce our treatment the wound burn, and then with the speed of the accelerate of the healing process. And it's very simple.
It's kind of in vitro, right?
In vitro? No,
in
in
silico.
animal.
Okay, in animal. So we can do that if you want. Okay, so from the start, I mean, Dr. Sandy is sharing his expertise in very, very very experienced. But you as a student, you can also doing a very small research, just like he said, if you are interested in stem cell. And yeah, if there's one more questions, if not yet, then I have a question. Oh, okay. Baikbal, you always have question, please.
Dr. Sandy, thank you for your insightful presentation. Actually, I have questions regarding in the topic of stem cell and secretome. Even through the GMP process, have you found inconsistency in the result during your stem cell therapy or secretome therapy? Because this is like not we control the chemical compound, but we control the cell, but not the behavior. So sometimes we produce the stem cell through the GMP process, but we cannot control the behavior of the cell, whether the cell want to produce more secretome, for example, or even though the cell produce more secretome, how about the enzyme inside the secretome? Is it the same? So if you have found any inconsistency in the result, how do you manage that inconsistency? I think that is my question. Thank you, Dr. Sandy.
Yeah, thank you.
Very practical question.
Yeah, it's very important thing because now we on progress to submit the registration to to BPOM for the secretome for the osteoarthritis indication. We hope in this month we can get approval for the clinical study for the registered secretome. And our experience to to discuss with the BPOM, yeah, BPOM always said the variability of the batch to batch concentration of our secretome, because the secretome part of the come from the stem cell. The stem cell is a live cell, yeah, and how to homogeneity of our stem cell. The important thing is we using the one donor, not the multi donor. So this is to prevent or to anticipate the variability of the secretome of our stem cell. We using a one donor from umbilical cord. We take the umbilical cord since 2013, and now we have around 5 trillion cell from one donor. This is to anticipate the variability of batch to batch. And the second, the medium you are using is the same, not different. And then the medium is animal free. There is no animal cell content in the medium. And fortunately, the result is still stable and is a homogeneous. And we can use the parameter of like growth factors concentration, like SGF, IGF, FGF, and anything growth factors, still in range, because it's a one donor, only come from one donor. And the important thing, like I think secretome like the herbal, because so many bioactive in secretome, yeah, and the BPOM always asks what the main bioactive in the secretome. Is very difficult because it's a very integrative interbioactive to give the result. If we take the one bioactive, maybe the result is no effect from the secretome. And so we propose to Badan POM, we do the potential assay, because the secretome has a potential assay, is a anti-inflammation. So we make it this assay to prove that the secretome have a potential anti-inflammation, anti-fibrotic, and so on. And again, for the minimize or to anticipate the variability batch to batch, we just use the one donor, and then the same environment for the stem cell.
Okay, thank you, Dr. Sandi, for your answers.
Yeah.
It's satisfying.
So according to your experience, there's no experience that the result is inconsistent, right? Okay. So if only if it happens.
It happened, we destroy it.
Okay. We destroy
Very
it.
expensive
Yeah,
actually,
it's-
but maybe before the destroy, I can give the best to the Pancasila University to do the research.
Okay.
Yeah, but I don't hope the
It happened.
It happened.
Because it's very expensive.
It's very expensive.
Yeah, that's right. And maybe is there any questions more for Dr. Sandi? If no, so I have, I really wonder about, you also have experience in other country, right, Dr. Sandi? And which country is the most advanced in this field and why? Maybe that's the first question. Yeah.
Yeah, I'm so many times present in each country abroad, and they do is very certified, very excellent in the research, because we know they have the equipment, the medium. Yeah, you know, if we order the medium in Indonesia, they around 3 or 6 months, and from Singapore, Malaysia, they just around 1 or 2 days they get the medium. It's very unfair, I think, yeah, for Indonesian research.
Yeah.
I think the research Indonesia is very clever or smart, yeah, but because the environment, infrastructure to support the research, and the important thing, they just do the in vitro and animal study. But in Indonesia, we can do the clinical study. So they always surprised for our presentation because they see in the human the result of the stem cell or secretome other than in animal and in vitro. Why Indonesia can do this? Because the Ministry of Health is very support to develop the cell therapy in Indonesia. And then now the Minister of degree of the Minister Health around I think the 2025, they allow to using the stem cell for the orthopedic indication and plastic surgery for the human. It's a standard treatment. Then so many hospitals now using the stem cell secretome for the orthopedic indication like osteoarthritis, HNP, yeah, and for the pain. I think it's very important for Indonesia. So many industry, stem cell industry in the world go to Indonesia now.
Wow.
Yeah, and then we compete very tight with the stem cell industry from abroad.
So we are moving forward, right?
Yeah.
We are moving forward. Okay, that's really good news, Dr. Sandi.
So we should from the academic using the stem cell, because now the trend is in the world is a stem cell, cell therapy.
Yeah. If I want to ask you about your perspective, how about the human resource that we have? Are we having good enough human resources for stem cell, or we
For
feel
human,
like
for human resource for the stem cell therapy.
Yeah.
Now, very difficult to find the good person in the skill, like a culture area or in cell. So we need so many students and like to enter the stem cell research or production, because now so many abroad industry stem cell bring the stem cell technology in Indonesia and make it manufacturing in Indonesia. I think in the future near future, I think.
Yeah.
So many industry in Indonesia, stem cell industry need the human resource
Yeah.
for the to do this.
Yeah, I hope, do you have further question, Pak Iqbal?
No, I think this is opportunity for Faculty of Pharmacy Universitas Pancasila to provide its students with the knowledge and skill and cell culture, I think. So hopefully we can propose this to our newly dean and get approval from her, for example.
Yeah, I think it's also a very good opportunities for our student and also for the Philippines, for everyone who is interested in stem cell, because just like Dr. Sandy said, we are moving forward maybe very fast, because it's very fast, and you can challenge yourself in this area. Okay, so I think that's all. If you have no further questions, guys. Okay, so Dr. Sandy, as we are approaching the end of the session, I would like to express our sincere appreciation to Dr. Sandy Klintang, Master of Biomedicine, for his outstanding presentation and also a very profound answer for this interactive discussion. I would also like to thank you, all the participants from Indonesia and the Philippines, and for your active participation and thoughtful questions. We hope today's inspires further collaboration between traditional knowledge and modern biotechnology in shaping the future of pharmacy. With that, I conclude this session and hand the program back to our Master of Ceremony. Thank you, everyone. Enjoy the rest of the International Summer Course and have a wonderful day. Thank you, Dr. Sandy.
Thank you, Dr. Sandy Klintang, for your wonderful presentation. We also sincerely thank Mr. Faradiba for moderating this session. So now I would like to invite Mr. Iqbal to give the certificate. For the documentation, committee, please take the picture in 3, 2, 1. Another one, Pancasila greetings. 3, 2, 1. Okay, thank you, Dr. Sandy and Mr. Iqbal. So before we move to our next agenda, I would like to announce the door prize for participant who actively answer the question. Actively ask the question in the first session and the second session. Okay, so from the first session, Safira Ramdania from FFUP.
Please come up to the stage.
Okay, the second one, Arvel Brian. Please come to the stage. And the third, John Ray Valenzuela. Sir Iqbal, maybe you can give the door prize.
Yeah.
No, no receipt.
Yeah. Okay, for the documentation team, please take a picture in 3, 2, 1. Another one, 3, 2, 1. Okay, next, let's take a picture in 3, 2, 1. Once again, 3, 2, 1.
Last one. Take picture in 3, 2, 1. Once again, 3, 2, 1. Okay, thank you for. You can take your seat.
Okay, thank you so much. Oh, another one, another picture. Okay, documentation team, 3, 2, 1. Another one, 3, 2, 1. Okay. Thank you so much for the active participate. Please be seated. For the second session, active participate. The first one is Francesca Alison Faith from Trinity University of Asia. And then the second one is Gerat Chan Tanda from FFUP. And then the third one is Takesha Julian from Trinity University of Asia. Congratulations. Please come up to the stage. Okay, for the documentation team, if you're ready. Okay, take a picture in 3, 2, 1. Once again, 3, 2, 1. Okay. And next one, 3, 2, 1. Once again, 3, 2, 1. And the last one, take a picture in 3, 2, 1. Once again, 3, 2, 1. Okay. Thank you so much and congratulations for the participant and please be seated.
Okay, before we move to our next agenda, attendees are invited to take a break for lunch and to pray for our Muslim guests. So our break session is scheduled from now until 12:50 p.m. So before 1 p.m., you guys have to come back to the auditorium. Okay, so you guys can break now.
Okay.